Currently, I'm working in Dr. Showman Lee's lab in year IMB, and she's researching something about chloroplasts in plants. So what we do is we want to know how the proteins get translocated into the chloroplast. You know, chloroplast is a double membrane organelle, yes. And most of the proteins inside chloroplast are made in the cytosol, which means that after it is made, the proteins has to be translocated into the chloroplast. In order to get into the chloroplast, it has to across the two membranes. So we focus on the things that happens in that double membranes. So there are two membranes. So you can imagine that if something is big, like a macromolecule, say a protein, has to get into the chloroplast, there must be a pore or a hole so that the protein can get imported into the chloroplast. We call those pores and other components helping those pores to translocate proteins. We call them translocomes. And we want to know how these translocomes work together to import those proteins into chloroplast. And so what we want to know and what I'm focusing on my graduate study is the mechanisms, the molecular mechanisms of these translocomes. Majorly focus on proteins level. So you ask me the scientific questions. So actually the most amazing thing for me currently is that for the chloroplast proteins to be recognized by those translocomes, there must be some specific receptors. Once the receptors recognize the proteins, it can be translocated to the chloroplast through a translocome, a pore. So I'm focusing on how the receptor protein recognize and bind to those chloroplast destined proteins. Yeah, so I do some biochemistry, try to figure out how these receptors bind to the chloroplast proteins and eventually translocate it into the chloroplast. But one thing I have to tell you is that the most amazing thing is that, you know, compared to chloroplast, cytosol is something very, very big. It's like a football field, as I told you before, right? Yeah. So a protein is kind of an ant. You put that protein in the cytosol, it's like put that ant in the foot field. So and you are asking the ant to find a particular exit or particular seat in that foot field. So it's very difficult for that protein to locate its destination. So the most interesting question for me is that how does this protein get transported to the surface of chloroplasts? Because it's a very long way, and a protein by itself, it's impossible to get there. So there must be something helping that protein. That's something I really want to know. Okay. I hope I graduated then.
I hope too.
Yeah. So in 10 years, I'm not quite sure that whether I'll stick to this career in 10 years, but one thing that I can tell you now is that I hope I won't lose the appetite for science.
Great.
Yeah, because as I told you, it's like say I like steak very much, but if you kept feeding me steak for 10 years, it's actually impossible for me to endure that. And someone could say, we have different ways to cook that steak. You can enjoy the difference of the flavors of your steaks. It's like you can enjoy all kinds of science. You can always find something different in science or in biology. But honestly, I believe that I will still love science, but I'm not quite sure that I'm not sure that I would love to try to stay alive, try to survive in the academic atmosphere. It's kind of difficult. So let's just say I will still love science in 10 years. I think everyone is busy, and the initial purpose of me trying to build up this kind of scientific discussion club is try to connect all the graduate students here. If you stay in your lab for 24 hours, say, or 30 hours every day, it's impossible for you to know people. So yeah, we try to build up the connections within the students. So how fun is it? It's very fun if we got enough people to join. So the main thing we do in that club is we have papers or some scientific papers you like or some seminars. rehearsals. So someone is going to present it, and all the fellow students give him or her our ideas about what do you think is interesting about the topic or how you presented it. So the fun part is actually, it's not really about the science so far. It's about how we communicate with each other, how we share our ideas. Within the lab, sometimes you may not dare to say something crazy. But here, in that discussion club, you can say anything you like. You can say anything about your imaginations. So that's the fun part. The people is the fun part. Oh, it was very good. And I think I was the only Taiwanese student in that committee. And last year we have Louise, George, Wendy, and Bernhard, and Emmanuel. Yeah. All international students.
Yeah.
So it was actually quite delightful for me. But inviting a big shot PI abroad to here to give a talk, all the procedure is complicated, and you have to do a lot of work to make it work.
Yes.
So I was the one who just did the easy part. Writing the invitation letters to the PI and organize everything is very different. It's very difficult, but I just did the easy part. But so far, I think it's very good, and it's actually a pleasure and my honor to work with you guys to make this happen. The most significant one, as I told you before, is that I don't need further trainings or help to read the menus in restaurants, so I can order by myself. That's it.